So today it's our great pleasure to welcome Shan Zhao, who will be telling us about the great things that you can do when you combine tissue clearing with light sheet microscopy and to map cell distribution in whole mammalian organs, which is a really interesting topic. So Shan has a background in pharmaceutical engineering, and she got her master's degree in applied chemistry at the Beijing Institute of Technology. And since then, she has worked in a number of different biomedical and molecular engineering projects. And then she joined the lab of Ali Ertürk at the Institute of Tissue Engineering and Regenerative Medicine at the LMU in Munich as a PhD student. She will tell us today about the exciting work that she's done during her PhD there. Thank you for joining us, Shan.
Yeah, thank you very much for the nice introduction. So then I will start. So good afternoon, everyone. Thanks the Euro-BioImaging group team giving me a chance to present our work here. So the title of my talk today is the holistic three-dimensional cellular mapping of the mammalian organs by tissue clearing and light sheet fluorescence microscope. So no matter we are interested to the human organs or animal model, the 3D map generation and the registration across the cellular and the spatial scales are very important for us to understand the physiological and also pathological functions. That's why the different countries across the world initiate different kind of projects, such as the Huber map, human cell atlas, human protein atlas. To achieve such long-term and communicated project, extensive knowledge and technology from different areas has been implemented. For example, using the MRI, we can check the gross view of the organ related to the structure and also identify the landmarks for integration. Then we can cut the whole organ into very thin sections. And for each of the thin section, we can check the anatomical character. We can study the specific structure using the histology. We can also investigate the high content single cell information when combining with the single cell analysis technologies. In the end, we can put all the data into the computer for analysis and annotation and to generate a high content three-dimensional molecule 3D map of the organs. But this map could be incomplete because of the cutting of the tissue. So we are also interested to provide an optional way to study the whole organ map by doing the whole organ labeling, whole organ clearing and imaging, and also data analysis to generate a very full continuously map. And after the organ recovery, we can still combine with the other technologies for further studies. So when we are dealing with the very large human organs, we know that the traditional histology method could be very low efficient. For example, for in intact adult human brain, it takes 6 months to cut it into 7,400 slices. And also take quite a long time for each of the slides. We do the civil staining, we do the imaging. So the whole process are quite time consuming. Very laborious, and also lack of the scalability. We cannot handle several organs at the same time. So also when we cutting the tissue into thin sections, imaging each of the section cannot provide enough information. So especially when the tissue distortion or they are losing of the tissue slice. That's why our lab and also the other labs are interested to do the whole organ clearing and the whole organ imaging. This technology is based on the refractive index matching mechanism. We can put the tissue into different kinds of the chemicals and make it become transparent. And then the light can pass through the organ inside and we can see the inner structures without section. So we hope this can provide a better model for better research. In the last decades, there are more than 40 different kinds of tissue clearing technologies are already published. And they are mainly classified into 3 categories: the hydrophilic method, the hydrogel-based method, and also the hydrophobic reagent-based method. This is also we are mainly interested to do. So when we collect a mouse or a sample, for example, here the mouse brain, we can optionally do the antibody or non-antibody staining, or we can also use transgenic fluorescence mouse lamp. And then we will put into the water miscible solvent to get rid of the water as the dehydration. And also put into the lipid miscible solvent to get rid of the lipid for the delipidation. So right now, most of the tissue is composed by the cross-linked or fixed protein. Then we can put the tissue into a media whose refractive index are the same as the cross-linked protein. Then, like the RR matching step, then the tissue can become transparent. So this method are very fast and robust. And also after the clearance, the sample are very solid, very easy to handle and imaging and also storage. And of course, because of the dehydration step, the sample also gets a shrinkage. And this, we think that is very better when we are dealing with the very large, the human organs actually. And also when you are using the transgenic fluorescence mouse lamp, probably the organic reagents could bleach the fluorescence protein. And that's why we are like optionally can do the antibody and the nanobody staining to keep it very longer time. So this comes to our main research aims. First part is related to the human organ. We want to develop a new tissue labeling and clearing method for intact human organs. And also we want to use light sheet fluorescence microscope to generate the 3D map. And when dealing with the very large size, the data from the large size, the human organs, we want to use deep learning method for fast and accurate analysis. And the second part is, of course, with the very wide used animal model, we want to develop a whole body of the mouse immune labeling method for reliable quantitative studies. And use this method, we can study different kind of disease models. So first part is related to the human organs. Recently, actually, there are scientists are very interested to use tissue clearing method to clear the human organs. For example, using the very famous and well-established clarity method, or people develop a new tissue clearing method, optical clear, by screening a lot of chemicals. Or we can embedding the human organs into the stretchable hydrogel, make it more stretchable and thinner. All of this method, we can see that we can handle a very small piece of the organ, and also it takes a very long time. So we are also questioning ourselves why to clear the human organs are so difficult compared to the mouse organs. There are very obvious caricatures, like the mouse organs are very small size, and also they are younger. They are like several months we will sacrifice them. But the human organs are very aged, and with the increase of the age, the lipofuscin also getting more and more. And also following the age of the organ, the insoluble collagen also gets increased. Also, naturally, the lipiderm of the human samples are much complex compared to the other organisms. And the fourth factor is that when we collect the human organs, because of the delay of the collection, so the human organs always carry a lot of the blood clot. We cannot immediately perfuse them like the mouse models. So the blood clots will cause strong autofluorescence and also impede the tissue transparency. Another challenge we are considering is how to label the human organs. The very advanced technology like the virus tracing or the transgenic fluorescence protein labeling method commonly used for the animal models, but cannot apply to the human organs. The human organs mostly rely on the post-staining method using the antibody or small molecule dye. We know that the antibody has very large size, and that they cannot penetrate deeply inside of the tissue. Even though we can clear them, but sometimes we cannot image high quality inner part of the tissue. And also for the small molecule dye, we lack some of the specificity, and also because of the natural color of the small molecule dye, sometimes the sample transparent could be impeded. So to address all of this problem, we developed the Chanel technology based on the small micelle mediated human organ efficient clearing and labeling method. So how is our hypothesis is that traditionally people will use the SDS or Triton to promimelize the tissue. And we can see that the Triton or SDS, they have the typical head to tail structures, like one end is hydrophobic, one end is hydrophilic. And when dissolved into the water, this will lead to the formation of very large sized micelle. And when the tissue when we put the tissue inside of the detergent solution, the micelle will get stuck on the surface of the tissue, cannot get inside of the tissue. So here we identified a very special detergent called the SHAPS. It has a very special chemical structure, like the steroidal structure. It has one hydrophobic convex side and a hydrophilic concave side. With this very special facial amphiphysility, so this detergent will form much smaller micelli when dissolved into the water. And when we put the tissue inside to this detergent solution, the detergent can easily and efficiently go inside of the tissue, promimelize the tissue. To prove our hypothesis, first we check the micelle size using the small angle X-ray scattering method. And from the data, especially the radius of the gyration with the micelli, we can see that the SHAPS forms much smaller micelle compared to the SDS and the Triton. And the second experiment we designed is to check if this promimalization could enhance the dye penetration. We put different group of the samples into different detergent solutions, SHAPS, SDS, or Triton, and then we incubate with the methylene blue dye. And after overnight, we cut the sample in middle and to see how the inner labeling efficient. And we could clearly see that only the SHAPS have a very homogeneous and full labeling of the methylene blue dye for these samples. And for the other two groups, because of the micelle only penetrate the surface part of the tissue, and also the dye only label the surface part of the tissue. So based on this dye signal, we can quantify the labeling efficiency. So next, we want to address the problem is to screen a SHAPS compatible chemical for bladder decolorization. As I told you that the human organs always carry blood and it will impede the tissue transparency. So the red color of the organ of the bladder is from the heme structure. And here we screened or we identified chemical candidates. They share the similar skeletal structures, the nitrogen, carbon, hydrogen structure. And our hypothesis is that this structure could interact with the iron of the heme as a ligand and then elute the iron outside of the heme, then destroy this structure and get decolorization. What we do is that we collect the mouse PFA fixed blood, and then we mix them with different chemicals, and then we centrifuge. There are two layers will form. The first layer is the supernatant, and also the second layer is the pellet. Here, if we only mix the blood with the detergent solution, SHAPS, Triton, or SDS, we could see that the supernatant are still colorless, and the pellet still red color. This means there's no decolorization effect. But when we mix the blood with our chemical candidate, or mix the blood with the chemical candidate and also the detergent SHAPS, we could see that the supernatant layer become like light or dark green color, and the pellet layer become colorless. This means very good decolorization effect. And we also can take out a small part of the supernatant solution to check the absorbance profile inside of the visible spectrum. And we can see that at the 600 nanometer wavelength, there is a new peak came out compared to the control group dye water. And based on this new peak, we can use it as a quantitative index to quantify the decolorization effect. And from the quantification data, we can see that when we mix the sharps and all the candidates, there is an improved decolorization effect. And also we can quantify the like which is the best concentration for the sharp solution. And from our candidates, we can see that there are several chemicals can really achieve good decolorization effect. But based on the price and also the available package on the market, we choose the chemical 7, the methyl diethanolamine, in our future experiments, the NMDA chemical. So after we identify this chemical decolorization, we also check it with the mouse organs. We collect the different mouse organs without perfusion. Then it contains a lot of the blood clot inside of the organs. And then we incubate with the different detergents mixed with this chemical 7 NMDA. And after several days, we can see that only the sharps group can achieve a better decolorization for all of these organs. So right now we addressed the polymerization, decolorization, and also all these problems. And the next question we want to address is that how to achieve deeper antibody labeling. So here, except at the first step, except we put the organs into the sharps and the NMDA for polymerization and decolorization. We also introduced 3 different kinds of pre-treatments, like the delipidation, the ECM losing steps to further make the extracellular matrix more loose for the antibody to get inside. Of course, very obviously, the delipidation step will try to get rid of the plasma membrane, the lipid on the plasma membrane of the cells. And also we use the acetic acid try to hydrolysis of the collagen by cleavage of the non-covalent interaction between the collagen. And also we introduce the guanidine hydrochloride, which will extract the proteoglycan of the extracellular matrix. So with all of these pre-treatment steps, right now the spatial interval of the tissue matrix are relatively loose and are accessible to the bigger antibodies, and also, of course, for the small molecule dye. So after this treatment, we can do the antibody labeling and do the traditional clearing using the organic method, and then ready for the imaging part. So to prove or test our protocol, we first tried a pig brain because it's very easy to available. We bought a pig brain from the local butcher, and after the dissection, we can see that the pig brain are full of the bladder on the surface and also inside of the tissue because we cannot do the perfusion. And after the sharps NMDA pre-treatment, we can see that there's a good decolorization, no bladder. And after the whole protocol, the whole Chanel protocol, we can clearly see the grid under the transparent sample to show the whole pig brain becomes very transparent. So with this very good results, it encouraged us to go to the real human organs. First, we test with the biggest human organ about the adult human brain. Here, to speed up the whole process, we want to take the advantage of the vasculature system of our human organs. We want to use a pump system to perfuse all of the regions through the carotid artery and the vertebral artery of the organs. And with this perfusion system, the whole process could be speed up. And here on the right side shows the whole human brain after the SHAPS and MDA pretreatment. We can see that the sample without any red color anymore. And also here these tubings is the connected we connect with our pump and also the carotid artery and the vestibular artery of the sample. So in about like 4 months of the whole process, including the pretreatment and also top 3 dye labeling and also clearing. So we make the first time we make the human brain become transparent. And we can see that condensed light could travel end to end through the whole human brain to show the transparency. So of course, not only with the human brain, also the other human organs, like here I showed you a human kidney. And this is the human kidney without clearing. And here is the tubing which we connected to our pump to perfuse. And after the whole process, we can clearly see that the sample become very transparent. And also some of the main arteries are already revealed inside of the organ. So not only with the human kidney, for other human organs like the lung, the heart, our pancreas, thyroid, or any kind of most of the organs we test in our lab, we can also make it transparent. So next problem we are thinking about is how to image the very big, large size the human organs. We know that the commercialized microscope, for example, the ultra microscope 2, they have a very fixed and smaller size about the sample chamber. Very obviously, we cannot put the human organs inside. We cannot image it. So to address this problem, we collaborate with the LaVision Biotech company to co-develop a prototype light sheet fluorescence microscope. This microscope is showing like this, and it has a very bigger sample chamber with like 25 by 9 by 7 centimeter size. And here we can clearly see that intact human kidney could mounted on the sample holder. And when we put the sample inside of the chamber and full of the bubble solutions, when we switch on the light, we can see that the light can pass through the very transparent sample to show the transparency. And with this powerful light sheet fluorescence microscope, we can image the very large human organs. So first here I show an example about the human eye we imaged. And this human eye we imaged here shows the iris, which is the very thin circular structure of the eye. And also this is the structure can identify our eye color. It contains a lot of pigments inside of this structure. And when we zoom into the details, we can clearly see like the cell nucleus related to the structure. And also we can image the cellular of the eye, which is the white part of the eye, and also the suspensory of the suspensory ligament of the eye. Here shows that how we image the whole eye from the top to down across the Z direction. So to show the transparency and also the imaging about the details. Not only with the organs, we also want to see that with this protocol, if the antibody can really penetrate inside and we can image it. So here I show an example is the IBA1 labeled microglia cells with a human brain cortex part. This sample is around 1.5 centimeter cubic, like a thickness, and also lungs and the device. So when we like a pre-treatment and also added antibody and then clear it, make it transparent and image it from the 3D reconstruction. And when we see the inner part of the tissue, we can see that the antibody, like the microglia signal with the green color, can really go inside to the sample inner part. And also we labeled the cell nucleus with the PI, which shows the magenta color. And when we zoom inside with a high magnification, we can see that the microglia cells with very larger and also ramified size in the green matter compared to the white matter part. And also when we labeled this sample, and after the clearing is very solid and very easy to storage, and it's very stable. After several months, you can re-image it again. And next is that we want to try to image some of the intact organs and also to analyze some of the quantitative studies. Here I show two examples. On the left part is the EGFP pig pancreas. This transgenic pig line, the beta cells express EGFP for the pancreas. And after the whole process of the Chanel labeling, and we image it with the very large size, the prototype fluorescence, light sheet fluorescence microscope. And we can see that when we zoom inside the image, we can see that the isolate could be a single cell or cluster of the cells. And based on the volume of the isolate, we can quantify the percentage of the isolate across the very big volume pig pancreas samples. On the right side is about example about the human kidney. This human kidney, we labeled the vasculature system with the dextrin. We labeled the cell nuclear with a topology dye. And also we imaged the autofluorescence at the 780 nanometer wavelength. And with this powerful light sheet, we can construct the whole organ about the human kidney. And when we zoom inside to the details, we can clearly see that the afferent artery and also the responding glomeruli. So based on this very high quality data, image data, we can quantify or caricature the cortex profiles related to the cortex zone wise, and also the glomerular size diameters, and also the afferent arterial diameters. So right now, all of this data actually for the quantification, at that time, we still use the commercialized software where, for example, ImageGene or Amaras for the analysis. First, when we handle this big data is quite slow. And also most of this method, this quantification method from this software, they are based on the filter-based filters. For example, threshold of the brightness of the signal. So to achieve a very fast and also accurate data analysis, we try to think about how to use the deep learning method to analyze the very big data from the human organs. Here, as a proof the concept, we our computer scientists in our lab, they establish this new convolutional neural network, and to training our original light sheet data. Here we imaged the cell nucleus about the human brain regions. And using this network, we want to do the detection and also segmentation and the counting about the numbers related to the image data. And here shows the very original light sheet data about the sample. And we can see that when we image this large size sample, we need to do a mass cycle imaging, and we can see some of the dark lines related to the mass cycle overlapping. So we can see that the image quality or the signal are not homogeneous. But when we put our data into our training network and to generate a binary data, we can see that after the segmentation, these binary data are very high quality, and we can identify the 6 layers about the cortex. So based on this deep learning based neural training network, we can detect and segment and also counting millions of the cells in different kinds of human brain region in several hours. So here shows, for example, the primary visual cortex, primary central sensory cortex, primary motor cortex, and the hippocampus. So with this part of data, that we achieve our main purpose related to the human organs. So let's go to the second part about the whole body mouse immunolabeling method and also for the disease model studies. So in 2016, at that time, our lab already developed the UDisco clear method, which can already make the whole body of the mouse become very transparent. But we can also see that not all of the parts are very transparent, especially like the bone marrow part. We can see that still red color, right? So, and when we image the inner signal across the whole body of this mouse, we can see that on the surface of the signal are still okay, but when we go deeper inside of the organ, we can see that the signal become very poor and very dim. So with this method, it's not will not allow us to do reliable quantitative studies across the whole intact imaging. So to address this problem, we developed this VDisco technology, which is based on the nanobody boosted 3D imaging of the solvent cleared organs. Here also we introduce a pump system, and we will deliver decalcification effect, decalcification regions like EDTA to make the bones get soft, and also we perfuse the decolorization chemicals to get rid of the remaining blood inside of the bone marrow or inside of the inner organs. And also very importantly, we perfuse the nanobody inside of the whole organ, whole body of the mouse. The nanobody has 10 times smaller size compared to the antibody. That's why it can penetrate inside all of the organs. And also this nanobody, we can conjugate very stable and very bright chemical fluorophores, for example, the Alexa dye or the Atto dye. So with this method, we can see that we can generate very transparent, basically like an invisible mouse and we can clearly see that the inner organs, even the bone marrow part, they become very, very transparent. So through this very transparent mouse, we can check the inner, we can easily check the inner signal from the inner part of the whole body of the mouse. And also because right now the nanobody conjugates very stable and bright chemical dyes compared to the a transgenic fluorescence protein signal is much stable, much bright. So the signal to background, the contrast become very, very high. So use this method, we generate the neuron connectivity of an adult mouse. This is from the Taiwan GLPM mouse, which the subtype of the neurons express GLP. And also, right now we can image the inner signal across the bone part, for example, through the skull. We can identify the skull meninges connections. Here shows the superior sagittal sinus, and here is the brain part, and across the sagittal sinus and the meninges, we can identify these small connections. And also due to the high quality about the signal, so we can right now do the auto neuron tracing. So let's go to the details. Here, when we put our nano boosting process compared to the unboosted sample, we can clearly see that the signal at the inner part of the brain or the cerebellum part, which contains a lot of lipids, the signal are much, much higher. Basically, we can enhance the signal intensity to 1 to 2 order of the magnitude. And here is the example. Sorry. the example about the Taiwan JLP mouse brain. This is like a same mouse brain, and then we cut into half. One half is unboosted and one half is boosted. So with this comparable experiment, we can clearly see that after the boosting, the signal gets very, very high quality and also very stable. After like 1 year, you can still image it. And not only we can clear like the inner organs and also the bone marrow, sorry, the bone part. Here, we can also imaging the cells under the skin. Traditionally, when people like clear the mouse organs, they will peel off the skin. And here, we keep the intact skin. And for example, as the Taiwan JLP mouse, and we want to image the nerves like under about all the organs. When we do the whole body imaging through this transparent mouse, we can see that the neuron projections coming out from the spinal cord, and under the skin, we can see the details about the neuron projections. And the green color shows the Taiwan JLP neuron signal, and the magenta color shows the PI labeled cell nucleus. Here shows the, oh, my mouse is a little bit. Okay, here shows the skin. Another disease example is about we also want to use this method to study different kind of disease models. Here I show the example about the TBI, traumatic brain injury. And for this disease, we study the peripheral nerve degeneration. For the disease model, we give a hit on for example, at the mouse left part, and we want to check the peripheral nerve degeneration across the whole body. So we identified that on the TBI disease model, after 1 month, we cleared the whole body of the mouse and we image it. We can see that at the upper torso part, the complexity about the nerve terminals at the neuromuscular junctions gets decreased compared to the control and lesioned samples. And also we can see that the neuron degeneration happened at the contralateral body side. And based on the axon endings quantification, we can see that there's a very significant degeneration related to the peripheral nerves. So next example is that we want to use this method to study the cancer metastasis and also the therapeutic antibody efficiency. So here for this, like we can transplant the cancer cells here, for example, MDA-MB231 cancer cells into the mouse, and after 2 months, the tumor metastasis happened to different parts of the mouse body. And optionally, we can also inject the therapeutic antibody into the mouse. And then we can do the VDisco whole body clearing and also imaging. And then we try to use the deep learning method to segment and analyze the data. And here we can see that with the very high quality light sheet microscope, we can identify the primary tumor and also the metastasis at different parts of the organs. For example, here the lung, the kidney, and also the liver. And when we also inject the therapeutic antibody, we want to see the colocalization between the cancer cells and also the therapeutic antibody signal. If we see there's a colocalization, there's a targeting effect. If there's only like a one channel of the signal, this means no targeting effect. So based on this method, we can like try to quantify or analyze the drug therapeutic efficiency. So with more details, that traditionally people will use the bioluminescence to like check the like inner organ signals. For example, here the tumor metastasis. But we know that this method are highly depending on the explorer time. When we have higher explorer time, we can see that when we have longer explorer time, we can see that the signal are basically everywhere. If we have a proper like explorer time, and we can see that the signal mainly focus on like the very strong area. So basically, we cannot really see if there are metastases really happened inside of different parts of the organs. So when we do the whole body clearing using the Vidisco and also light sheet microscope, basically we can identify single metastasis. Here also, this is the results, the same like MDA MB201 cancer cells, cancer metastasis mouse. And also here we can clearly see that this is the primary tumor, and also on the other parts are the cancer metastasis. And when we see the results across the Z direction, we can see that at each plane of the samples, we see that the signals in the lung and also in the liver and also kidney parts, and actually also here at the axillary lymph node part. So basically, we can see that actually the metastasis happened inside of your very deeper inner part of your organ. When we are using the bioluminescent imaging, this kind of signal are some kind of very difficult for our to judge they are right or not, right? So with our method, we can clearly see that this really happened here at a metastasis. So of course, when we image the whole body of the mouse, it generates a lot of data. In general, like one whole body of the mouse is like 2 terabytes of the data. And also for us, it's very difficult to analyze the data using the commercialized software. That's why our computer scientists, they also develop a convolutional neural network trying to detect all the metastases using the deep learning method. And here we can see that using the deep learning method is quite fast and also accurate. We can basically count like how many metastases happened in different mouse organs. For example, the brain, the lung, the kidney. We can see that most of the metastases happened inside of the lung. And also same, this is the MDA MB201 cancer metastasis models. But also we did other like a cancer mouse lung, like the pancreatic cancer lung or the lung cancer samples. For different cancer lungs, using our method, we can see that the metastasis happened like the ratio related, or the percentage of the metastasis in different organs are different actually. On the figure D, we can see that across the Z direction, the depth of the signal, how many of the metastases happened. So not only with the metastasis, we also want to use our like neural network to analyze the drug delivery efficiency. Here is also a mouse model with the MDMB201 primary tumor and the cancer metastasis. And also after 2 months of the metastasis, we also inject a therapeutic antibody, the 6A10 antibody. which is targeted the CA12 inside of the cancer. So here we use like the whole body transparent VDisco technology to boost the tumor cells in one channel, and also the therapeutic antibody we conjugate with alicizadine in another channel. We want to see if there are colocalization in these two channel samples. And when with the 2D fluorescence microscope, we can already see that the tumors here shows with the magenta color, and the antibody signal shows with the cyan color. We can see that there are colocalization related to like the lymph nodes, and also in other parts about the lung. But we also identified some of the metastasis without any antibody signal. That means this metastasis is not targeted by the therapeutic antibody. So when also we further confirmed the same results with the 3D light sheet microscope, here shows the heart part and surrounded with the lung. Here is the axillary lymph node. And we can see that the colocalization in these two channels. So based on all of this high quality data, and then we analyze this data with our deep learning neural network, and we can basically quantify like how many metastases are targeted by the therapeutic antibody. And we see that there are like 77% of the metastases are targeted, and there are still some parts are not targeted. So with all of the data, I hope that I convinced you that we combine multidisciplinary knowledge, try to study the holistic 3D cellular details about the mammalian organs using the tissue clearing method and also imaging method. And then we try to use our method to study some biological applications. First, we developed this Shan Zhao technology for a robust tissue clearing and also labeling method. And we use a powerful light sheet microscope, can do the 3D reconstruction about the organs. And then using the deep learning method, we can also do a fast and accurate data analysis. And for the widely used mouse models, we generate this like super transparent mice with immune labeling by VDisco technology. And use this technology when combining with the deep learning method, we can achieve reliable quantitative study in a very fast and accurate way. And also use this method, we can study different kinds of disease models. So with all of this work, I would like to thank my supervisor, Dr. Ali Ertik, thank all of our lab members, and especially the one that we work together in this kind of project. And also thank all our collaborators, which like provide, for example, the human organs, the pig organs, and also support with the imaging part. And also thank our CSD building related to the animal facility. So thank you all for your attention. I'm happy to take questions.
Thank you very much for this presentation. Super fascinating work. The pictures are incredible, and the invisible mouse blows my mind every time I see it. It's amazing what you can do with this. Do we have questions from the audience? You can either raise your hand or just unmute yourself and ask your question. Okay, maybe while I'll give people a chance to think of some questions. I have the disease model part in the second part of your presentation with the mouse models. That made me wonder how scalable this is, because it still sounds like it's quite a lot of work and like working with these sort of so big animals and like all these things. But is this a thing where you could imagine doing almost like a screening approach already? Is this scalable, or is this still more of a, you do one mouse at a time and then that's it for the next month?
Yeah, that's a very good question. Actually, related to the like for example, for the disease model, in general, we will do like 3 at least in each of the group, right? Especially for the quantification data. So when we do when you group your samples and then you do the surgery, there are no problems. And for the clearing parts, also no problem, because our pump has like 4 channels, like one pump you can perfuse 4 mouse at the same time. So basically we have a bench and we have like more than 10 pumps, so we can handle one time like 20 mouse, there's no problem. Basically, you just set up the pump, and in between, you just change the solutions for different chemicals. So there's not so much about the lab works. The main limited step is, you are right, is like the imaging part. So before we use this new light sheet fluorescence microscope, it's quite difficult for us to image, because you see that even though the ultra microscope tool has a very small chamber, obviously the mouse cannot put inside. At that time, we need to fold the mouse into a very unnatural structure and try to image the belly part first, and then cut it and then image the other part. In the end, we like to fusion all the different parts together. It's general like to image one mouse probably take like 1 month. And right now with this new light sheet, basically the whole body of the mouse can easily mount on the sample, and we can image it one time. So basically right now, like we will mount the one mouse and the image on one side, and then flip up the samples and image the other side, and then fuse them together. So right now, imaging like one mouse take like 3 to 4 days. So basically, it's already greatly, reduced our lab work, and also faster the whole process right now. But of course, right now the limited things is that also, the computer handle this kind of big data. We save it and we analyze it. Also takes time. Yeah. So for the clearing part, no problem. We can do a scalable experiment. But for the image part, you need to image one by one.
Yeah, and then these are very different challenges from what microscopes are usually designed to handle in terms of the sample. So I can imagine that this new microscope design makes a big difference there. We have a question in the chat where somebody asked, did you identify the nature of the bridges between the skull and the meninges?
The nature?
You showed the nature of these bridges between the skull and the meninges. Did you identify what these are?
Yeah, I know that actually this is also a big project we are doing now. At the time, we identified this kind of small connections, and then at the same time, there are several other paper, like Nature paper workouts, that they identify this kind of small connections, and that people trying to study if these connections play a very important functional way, or like a structure related position related to the disease or the functions. For our lab right now, we still think that it's not some kind of like a vascular system. We identify some immune cells inside of these connections, and right now we still don't know what they are really is. This is one of the bigger projects our PhD and the postdoc, they are working together, like try to identify this kind of nature things, and also identify the functions. But right now, based on our knowledge, we cannot still like say it very correctly what they are. Yeah.
Yeah, but I think this shows the strength of this technology. It's opening up so many new questions.
Yeah, exactly. That's why we want to use this technology. I know that in the other labs, they like dissect the sample, try to peel off the intact meninges, and then image it, and to identify, to label with like a level 1 or any other like immune cell antibodies, like to study the functions and also, the nature structural things. for us, we are also trying to do this without dissection. Yeah.
And there's another question in the chat. The question is your deep learning based on an open source software platform?
Yes, all the work I presented already published, and all the codes and all the data, they are open. Yeah, they are opened, and you can check the original paper and try to see that. Yeah, or you can contact me, email me. I will send you the link. Yeah.
That's great. Thank you very much. That's awesome that this is available. Are there any further questions? Georgina, yes, go ahead, please.
I really enjoyed your talk. It's amazing. Thank you. It was just a point because there was a talk on the meeting I was at earlier in the week that there is a Slack channel for the light sheet community that it had about 8 people on Monday, and now it's got about 30 people. So people just wanted to make people aware of that. I thought that might be a good place to announce it. So I'm going to put the link in the chat, and I think they're really open to people joining. They've called it Wild Sheet.
Okay. Yeah, this new light sheet right now is already commercialized as Ultra Blaze, and right now the Laverian company is combined with a Meltony company actually. Yeah, that's why they commercialize it already.
That's great. Thanks, Georgina, for putting the link there for anybody who is interested in talking more with experts on light sheet microscopy. Are there any other questions? No? Okay, well then I would like to thank Shan again for this fantastic presentation, and thank you all for attending, and I wish you a nice Friday afternoon and then a good weekend. Bye-bye, everybody.
Bye-bye. Thank you.